Hello, my name is Juannan Zhou. I'm currently a postdoc at Cold Spring Harbor Laboratory. Today, I will talk about our method for modeling high-order epistasis in sequence function maps. So how sequence determines function is a fundamental question in biology. In recent years, high-throughput phenotyping assays, including massively parallel reporting assays and deep mutational scanning, have started to allow us to measure the function of thousands up to millions of variants in a single experiment. So these experiments enable us to study empirical sequence function maps or fitness landscapes for the first time. So one very useful way to think about this type of data is that the sample sequences are all from the space of all possible sequences, or the sequence space shown schematically here in this figure drawn by Sibyl Wright in 1932. So a major challenge in using these experiments to study fitness landscape is that the size of the space of all possible sequences grows exponentially with sequence length. So in most cases, we will be able to measure only a tiny fraction of the fitness landscape. So there is almost always a need to use computational methods to predict the function of unsample variance. So predicting the function of novel variants is a hard problem because mutations often combine to determine fitness in a non-additive manner. And this phenomenon called epistasis can occur among any number of loci, which makes the prediction of novel sequences very difficult. A common approach to modeling epistasis in a regression framework is to add additional terms on top of the additive terms to account for pairwise, sometimes three-way interactions. So the problem is that the number of terms grows exponentially as the order increases, making this unsuitable for modeling high-order epistasis. So the problem we try to solve here is first to understand the complex pattern of genetic interactions in the data, and second, how to use this understanding to make better phenotypic predictions. So the solution we propose is to first learn the type and extent of epistasis using the so-called variance component analysis, and then to reconstruct the sequence function map using the inferred variance components with Gaussian process regression. So what are variance components? They are a list of numbers that correspond to the fraction of the total variance in the data that is explained due to each order of genetic interaction. So shown here is one such example for a fitness landscape for sequences with 8 nucleotides. So why is the variance components an important statistics to learn? It turns out that it encodes a lot of the information about the structure and epistasis of the fitness landscape. So first, the variance components determines how the correlation in phenotypes decays as a function of sequence divergence. And second, the variance components also determines the distance correlation of mutational effects and local epistatic interactions. So these quantities are all extremely important when making predictions, as they tell us when we see a mutational effect or a local interaction, how predictable their effects will be when they occur in a distant genetic background. So how do we infer the various components from the data? So our method here relies on a mathematical result that says the distance correlation of phenotypes when there is only a certain number of interaction takes a very specific shape, as shown here on these two panels. So given an empirical distance correlation shown here in the black, we can identify the best combination of these curves that gives rise to the observed curve, and the weights for these curves then correspond to the variance components. So with the learned variance components, we can then encode it probabilistically into a Gaussian process prior, so that a typical sample from this prior has the same statistical character as the data. We then use Gaussian process regression to make predictions as well as to assess predictive uncertainty. We also came up with a couple of computational tricks that allows us to perform Gaussian process regression efficiently using sparse matrix multiplication and efficient posterior sampling using Hamiltonian Monte Carlo. So we then apply this method to a high throughput dataset that measures the splicing activity of human 5' splice sites. So the 5' splice site is an 8 nucleotide long sequence, so the size of the sequence space is 4 to the 8, which is roughly 65,000. So on these two panels, we have the distance correlation of phenotypes and mutational effects. And we know that they deviate quite strongly from the additive and pairwise assumption. And this is because the splicing landscape contains substantial amount of high-order epistasis, as shown here by the variance components plot. So our model also shows superior performance in terms of out-of-sample R-squares with varying degrees of training data density, as shown here, compared with popular methods for modeling sequence function relationships. Last, we present a visualization of the splicing landscape based on the reconstruction by our model. So here we show that the structure of the splicing landscape can be largely understood in terms of specific patterns of epistasis, including some novel genetic interactions. So to conclude, we find that variance components provide a high-level summary of the structure and epistasis in the sequence function maps, and incorporating the empirical variance components can greatly improve our predictive accuracy. With this, I would like to thank my co-authors and funding sources. Please find the links to the preprint as well as the software. Last, I'm starting my own lab at the University of Florida this coming fall, and I'm currently hiring postdocs, graduate students, and lab techs. So please send me an email if you are interested. Thank you for your time.